Namaskar दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing unlike any recertification question, DABT question, we'll discuss important topic with respect to principles of toxicology as well as point of departures because it is an important in the case of ABT brochure syllabus they have given and also try when I have written the exam in year 2023, there were questions on. They used to give pictorial, different types of dose response curve they used to give and would give certain options to interpret that particular graph. Sir. Okay, so let's directly see the topic. So first we'll discuss about the different dose response curves. What are the point of departures one can get from this typical repeat dose toxicology study? Once we plot a graph that is called dose response curve. So what are the different point of departures we have? So first let us understand. So in the as a toxicologist or pharmacologist, you'll come across different types of curves like dose response curve you'll come across. In case of inhalation, you will come across exposure response curve. Okay, and in the case of kinetics, don't get confused. Concentration response curve you'll be having. Okay, so that and this is little bit of different. From that concentration, in the case of kinetics, you will be getting AUC, Cmax, and Tmax. Okay, so compare and don't get confused. So let's directly understand the first dose response curve or exposure response curve describes the relationship between the amount of a test article and the incidence or magnitude of a biological effect. So once to a test system, test animal, if you give a test item, so how that particular animal is giving a response based on that incidence or magnitude of the biological effect. The biological effect might be change in the clinical parameters or appearance or physiological response like that. They are fundamental for determining safe starting doses and identifying the potential risk. So if you see a typical dose response curve, no, on x-axis you will have dose which is in the units mg per kg. Okay, so whenever you are giving the test item in the form of oral route, you will be having units in the form of mg per kg. And whenever you are working with an anti-cancer drug or you are applying the test item through parenteral route or through dermal route, the units will be in mg per meter square. So these units also you have to always remember. And on the y-axis you have response. Response may be in the form of percentage or in the case of in the form of magnitude. So we'll see three different types of dose response curve. Here, if you see the red color, no, it is adverse. This red color graph shows adverse or toxic response, and the blue color is therapeutic. So the pharmacologist will focus more on this particular blue color line, that is therapeutic beneficial response. As a toxicologist, you will be looking at adverse toxic response because from this only this adverse toxic response you'll be getting NOAEL as well as LOAEL. Whereas therapeutic beneficial response, pharmacologists will get ED50. And third one, which is a green one, no? This is called threshold type or non-monotypic response, from which you are going to get your toxic effect, median toxic dose. Okay, so first one we'll see. If you see, first we'll understand efficacy curve. As the dose is increasing, no, the response is increasing. So this is blue color is expressing about the your therapeutic efficacy curve. So here, if you see on this blue color, 50% of the response, you have to take it from the y-axis and extrapolate it to the to your efficacy curve. And when you extrapolate down to x-axis, no, this is the dose. This is the point at which you will have median effective dose, ED50. Okay. So similarly, in the case of adverse effect dose response curve, if you see here, taking 50% response from the y-axis and extrapolating it to the this adverse effect dose response curve and intersecting at this particular point, and when you move this particular line towards x-axis, you will be getting your 50% of the your response, toxic response. Okay, so if you see this adverse toxic response curve, no, here this is the point one. Point one is the dose at which you will be having adverse effects. For example, you're working on a certain drug and it will start showing some adverse effect on the kidney or liver. So this is the point at which it is showing, uh, uh, it is not showing any adverse effects. It is similar to that of the control. Okay, and here if you see, this is the point at which it is showing adverse effects. This is called lowest observed adverse effect level. So any at any point on this curve, any single point above this curve will be considered as causing adverse effects. So it might be 50, 51. I mean to say it will be 1, 2, 3, 4, 5, 6 like this as the dose increases. Okay. So what you have to remember here is L LOL. That is, you have to understand that lowest dose at which observable adverse effect is observed. So first we will see the definitions very quickly. NOAL is the highest dose at which no adverse effect is observed. Okay. LOL is the lowest dose. Okay. Here you will see 1, 2, 3, 4. As the dose is increasing, effect is increasing. So out of which one can be LOL, 2, 3, 4. All these doses are causing adverse effects, but one will be the lowest dose at which adverse effect is there. Okay, and ED50 is the or this blue dose response curve, dose producing a therapeutic or beneficial effect in 50% of the subjects. And coming to this one, this is your threshold type or non-monotopic dose response curve, which is giving your minimum toxic dose producing a toxic effect in 50% of the subjects in that particular study. Okay, so one question in the ABT you'll be getting with respect to this therapeutic index. Okay, it is there in the syllabus. So they will try to confuse you. They'll try to give the options close by so that you'll not be able to identify the answer. Generally, the answer is therapeutic index is the ratio of this TD50. From this, you are getting this TD50, no? TD50 to ED50. This is your therapeutic index. So higher the therapeutic index indicates a wider margin of safety. So in between these dose response curve, no, the distance between these curves should be as much as greater if it is good. Why? Because physicians will have a chance to play with the molecule. For example, in the case of digoxin, where you have narrow therapeutic index, then you have to do continuously monitoring of this particular drug, plasma monitoring of this drug you need to be followed. For that reason, wider the margin of safety, it is better the safer the molecule. Another thing that you have to remember with respect to another word, your key term you'll come across is potency. As you move towards the left side of the curve, okay. So lower the dose, more the efficacy. That is called potency. So left shift is more potent, more response at a lower dose. So same drug, for example, there is a drug A which is showing effect at 1 mg per kg, and another drug B which is showing the efficacy at 10 mg per kg. Both belongs to the same class of drug. So physician will go for the potent drug. That is, if you're taking less amount of drug and it is able to give the same efficacy, you go for higher potent drugs. Okay. So what are the new drugs which are coming into the market? No, they're almost all are highly potent drugs. Okay. If you see the older traditional drugs, they used to be 100 mg, 500 mg, the dose strength, if you see. But today the doses are in like micrograms. But this itself is having a very great challenge with respect to manufacturing challenges in there. Handling the potent drugs is also a major threat to the human population. Key takeaway that you have to remember is dose response curve help identify the relationship between the dose and effect, supporting selection of the safe starting dose, because from this only we'll be able to understand how to consider the first dose, first in human dose in the case of phase 1 trial for clinical trials and evaluation of the potential toxic effects. If we understand, okay, this is at the target organs, then this adds help us to monitor, consider these points in the case of when you are doing phase 1 trials. Okay, important consideration. Curves are typically generated from repeat dose toxicology study. So, in the case of a single dose toxicology study, you will not be able to develop the S-shaped dose response curve. Okay, you have different type of that Litchcoxing method or Wilcoxing method will be there from which you can derive the LD50, but you cannot derive S-shaped dose response curve from the single dose tox studies. And important, another thing is shape and position of the curve may vary by species. So if you're taking a single study, you have single dose response curve for a particular effect. So rat will might be different, mice will be different, rabbit, monkey, and dog. And shape and position of the curve may vary with the species, sex, and the study design. And risk assessment uses NOEL and NOEL for the appropriate uncertainty factors. So once you get NOAEL or NOEL, you can use it for risk assessment or deriving certain health-based exposure limits. So now we'll see what is the difference between NOEL, NOEL. So I'm not remembering, you go back and check when was actually GLP started. Maybe it was in pre-GLP, like I think GLP 1920 or 1930, something like that. So before GLP, if you see those kind of studies, more than NOEL, you will come across this point of departure NOEL. And if you look into ICH Q3C guideline also, where you're deriving the permitted daily exposure for certain solvents, you will be getting point of departure as NOEL or NEL, okay, rather than NOAEL. So we'll see. So how a toxicologist will have a challenge when he is doing literature, search, how whether he will consider NOEL or NOEL, and if we end up with the NOEL or NAEL, what to be considered, that we'll see. Okay, little bit of differences we'll try to learn. So difference between NOEL and NOEL in toxicology, both are derived from the dose response curve and refer to doses below which no adverse effects are observed. So NOEL is the highest dose at which no biological effect, whether it is beneficial or whether it is adverse effects, they should not be similar to that of the control. It should be no biological effect of any kind, adverse or beneficial, is observed compared with the control. Okay, coming to NOEL, NOEL is the highest dose at which no adverse effect is observed, beneficial, that is positive effects, or it may be present. Okay, so this is in the previous slide, as we have seen, S-shaped dose response curve. If you see on the left side, this is a flat line is there. So on this flat line only, you will be having NOEL as well as NOAEL. Okay, so this is your adverse effect toxicity, no adverse effect, and no effect. And this green line, it indicates this is the point at which there was no beneficial or adverse effect, and NOEL is also is the point at which uh, 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 both the beneficial as well as positive effect. So certain times, this NOEL and NOEL may be equal, and certain cases NOEL might be greater, compared is equal or greater than NOEL. Okay, so we will see the comparison NOEL, NOEL, and the key differences between these two point of departures. No effect of any kind, neither harmful or beneficial, compared with the control. No adverse effect, harmful effect, beneficial effect may be present. NOEL is stricter. So once you come across NOEL as well as NOEL, NOEL is more stringent. Okay, NOEL is specific to certain adverse effects like kidney effects or liver effects like that. Indices includes absence of both adverse and beneficial effects. Okay, if there are changes in certain body weight or color changes or fur changes, those also. Okay, and focus that also need to be considered as an effect, and that particular dose also should not be considered as an NOEL. Okay, focuses only on absence of adverse effects, beneficial effects not excluded. NOEL exclude any change, and NOEL excludes only harmful effects. If there is any change in the body weight, NOEL will ignore. Okay, they will only consider a serious type of adverse effects. And also, it also depends on the study director also. Certain times, case by case it is. And position on the curve, left most dose on the flat baseline. This baseline I told you, no change from the control. This is your control, this is your NOEL, and this is your NOEL. And or the right NOEL still on the flat baseline, and NOEL is equal to or greater than. This is the important thing that you have to remember. Certain times when you do the literature search, you'll come across NOEL equal to or greater than. So you have to be very careful what you're seeing. Those symbols are also very, very important. Considering the application in the case of risk assessment, NOEL less commonly used today. As I told you, if you see the pre-GLP studies, in that type of studies you'll be coming across NOEL. Or certain times in the case of that NTP type of study, not NTP, that 2-year carcinogenicity studies are there, in that you'll come across this NOEL rather than NOEL. Coming to this NOEL application in the case of risk assessment, yes, for the derivation of HBL, that is health-based exposure limits, you need for deriving RFD, for deriving ADI or occupational exposure limit, you need this NOEL as a starting point. Then only you can apply it to there are certain formulas, applying the uncertainty factors and deriving the limits. Regulatory guidelines recommend NOEL. So whenever you have an audit, if an FDA inspector or Canada inspector or WHO inspector is coming, you will be asking, if you're doing certain risk assessment, you will be asking, what is the point of departure? What is the source of, from where, what is the reference? What is the study they will be asking? What is the point of departure they'll be asking? So you have to be very careful. They always prefer that the whatever the safety limit is there, it has to be derived. The starting dose should be NOEL. They don't encourage NOEL most of the times. If nothing is there, they'll accept it. But the first preference will always be NOAEL. Okay. Now next, very quickly we'll understand what is the difference between NOEL and control. Okay. Because as a toxicology, I had a doubt in the beginning of my career. if both NOAEL and control are almost same, where it is not at all showing any adverse effect, why we need to have another term called NOAEL? Why can't we control NOAEL as well as NOAEL? So that we'll try to understand. So is NOAEL and control same in the toxicology dose response curve? No. NOAEL and control are not the same. Okay. Control is untreated. In the case of control group, we are not giving any test item. Okay. Used as a baseline for comparison. So anytime any treated group, if you want to compare, you need to have a control group. Okay. NOAEL is the highest dose greater than 0. Okay, 0 increase like 1, 2, 3, 4, or 5, 10, 50, 100 like that, at which no adverse effect is observed. So response, if you see, this is the adverse effect toxicity, no adverse effect, no effect baseline. This is the baseline. The previous graph you have seen only 2 points on this flat line, no? NOAEL and NOAEL. Here we have purple color, that is control. Untreated group used as a baseline for comparison. So we'll very quickly, and these are the green is NOAEL, blue is NOAEL, and LOAEL. Okay, so from the single dose response curve, so much of information you are able to get, and this information can be further used in the case of risk assessment. So then we will see the term control is 0, untreated group. NOAEL is highest dose, greater than 0. Okay, and represents natural background response used to detect any change caused by the treatment, determined from treated groups located to the right of the control on the dose axis. Okay, so control is a baseline, NOAEL is a treatment dose with no observed adverse effect. So from this, we will understand about the difference between NOAEL and control are different and represent different concepts. This is the take-home message that you have to remember. Okay, so this will be very useful in the case of risk assessment whenever you're doing literature search. Okay, very quickly, I will not explain too much over here because till now dose response curve, NOAEL control, the NOAEL we have understood. Now, what I would request is few questions on MTD as a risk assessment also you'll be coming across. So MTD is called maximum tolerated dose. As a toxicologist, I also had a very confusing thing that if you look into single dose toxicology, no, the main objective is to derive LD50. That you are getting it from OECD guideline 420, 423, and 425. Previously, I told the study secrecy 401 was the guideline. Objective is to single administration deriving LD50. But if you see nowadays literature search, no, in the single dose toxicology study also, in addition to LD50, certain times which will be having range, you'll come across MTD term, maximum tolerated dose. And there are certain studies like 3-day studies or 5-day study, which might be dosage finding studies or single dose studies, you'll be again having MTD. Okay. And the same MTD term, certain times you'll come across in the case of 28-day study or 90-day study, in which in addition to NOAEL and NOAEL, study director also reported maximum tolerated dose. And same term MTD will come across in the case of cancer study also, where this MTD will become the basis for defining the doses for the 2-year cancer study. So you can pause the video and see the comparison. Very quickly, important points we'll discuss. So purpose is in the case of single dose toxicology, MTD first you see the highest dose level that does not cause unacceptable toxicity or mortality. Okay, and is considered tolerable for the planned duration of the study. You see, in the case of single-dose toxicology study, LD50, where mortality is a checkpoint, is a major identification. In the case of MTD, we're pushing the animal to the maximum toleration, but we are not causing. It might move the animal to a morbid conditions, but we are not killing the animal. Okay, to identify acute toxicity and lethal dose range from a single administration. To evaluate systemic toxicity. So repeat dose, where objective is to find out if there is any systemic toxicity from the repeated administration over a short to intermediate duration. In the case of cancer study, to evaluate carcinogenic potential of the test article for a long period, that is up to 2 years. Here, if you look into the duration, one-time administration, and it might be observation is up to 14 days. Here, typically 14 day, 28 day, or 90 day up to 6 months. Here, 2 year study. MTD definition. This is important. Important thing you have to know. Highest dose that does not cause mortality or untoward effects. You need to understand what is this untoward effects within the observation period. Okay. In the case of repeat dose toxicology, same MTD definition is highest dose that does not cause unacceptable toxicity. Here, untoward toxic effects and mortality did not cause mortal death. It should not cause death, affecting survival, body weight, clinical size, and target organ toxicity. Coming to cancer study, highest dose that does not cause excessive toxicity, unacceptable, untoward, excessive toxicity, significantly reducing survival and is considered tolerable for a longer exposure period. So you can pause here and see what are the major basis for MTD selections: mortality, clinical signs, body weight, gross necropsy finding. We're not doing histopathology here. Survival, clinical observations, body weight, food consumption, hematology, clinical chemistry, organ weights, and histopathology. Here you are performing histopathology as well as organ weights. Gross pathology in the case here. Same, survival for 2 years, body weight, food consumption, clinical observation, hematology, clinical chemistry, incidence of non-neoplastic lesions, very, very important to identify the cancer, and histopathology. Okay. Typical MTD level usually limited by acute lethality or severe acute toxic effects, usually less than or equal to dose causing minimal but acceptable toxicity. This term is very important, less than or equal to. They are also usually less than or equal to dose causing severe or life-threatening toxicity. Animals should not die here. Okay. So hope this was a very interesting topic, I guess, for you to remember and learn, because it will be very challenging when you are working as a risk assessment toxicologist. Key point once again we see MTD is a study specific and duration dependent. Longer the exposure of the duration, lower the MTD is likely to be. MTD is used to set high dose level in the study and ensure adequate safety margin for the risk assessment. As I said, no, it is very important in the case of risk assessment toxicologist. So hope from this video you are able to learn about few concepts with respect to point of departures, which are given in the ABT brochure. That is NOEL or LOEL, NOAEL as well as MTD. Okay. Thank you so much. Be curious. All the best for your exam.